So people that have two personas. I'm listening.
Okay, alright. Chapter 1 is anatomy. So, what are the sections of the gut?
Head gut, forward gut, mid gut, hind gut.
Good.
Why did that sound like Dr. Seuss?
That kinda did.
Left foot, right foot. Red fish, blue fish, mid gut, hind gut.
Okay, anatomically, what is in the head gut?
The oropharynx.
Uh-huh.
Oral cavity
Yes.
and the esophagus.
Yes. And the pharynx.
Well, that's
Not the
all.
esophagus.
Okay.
What is in the foregut anatomically?
That's esophagus and stomach.
Yes. Who is in the midgut?
Small intestine.
Good. And hindgut?
Large intestine.
And
Colon.
this large intestine.
Those are part of the large intestine.
What else?
Anus.
There you go.
An anal
Rectum.
canal.
Perfect. Okay, so this is anatomically what you need to know for the sections. What about embryologically?
Oh man, can I try this?
Yeah.
Alright. So ectoderm.
Who's ectoderm?
Ectoderm is gonna be the head gut and the hind gut.
Is it?
No. The head gut and the last thing that is there.
Mhm.
The
The external anal sphincter.
is that it?
Mhm,
Okay.
basically.
Yeah, external anal sphincter and everything else is endoderm.
Is what?
Endoderm.
There you go.
Yeah.
So, pharynx is embryologically part of the foregut.
It is. So, what is our head gut embryologically?
So, the cavity,
Distal anal canal.
Mm-hmm. What is the foregut embryologically?
Everything else.
So it's going to be,
pharynx,
Oh, I'm sorry.
Yeah.
I thought you were like. So the foregut is the pharynx,
to what?
the proximal duodenum.
Perfect.
My question was, is the pharynx ectoderm?
No.
Okay, that's why I was asking.
No, it's not.
Okay,
I would count it as endoderm.
okay.
Okay. Because this is like anatomically how we separate it, and then embryologically where it develops from.
Right,
Yeah. Is that
Yeah.
okay?
Yeah.
All right. So who is midgut embryologically?
The distal duodenum to the proximal transverse colon.
Good.
Now, does distal mean after the papilla?
Yes.
Yes.
Okay.
To proximal transverse colon.
Stupid proximal transverse colon.
Oh, sorry. And then what is the hindgut embryologically?
Distal transverse colon.
Mm-hmm. To perfect. Okay. What artery supplies the foregut?
Cilia.
Cilia. Yep.
Cranial caudal.
Yes. Cranial caudal what?
Mesenteric.
Okay. Then you have, do we do esophaguses? Not really. Okay, so there are two types of stomachs. What are they?
Complex and simple.
Who has a simple stomach?
Carnivores.
Okay. Who else?
People. And pigs.
I believe pigs, yeah. And who has a complex stomach?
Ruminants.
Yes.
Pigs have a intermediate. Oh, that's colon.
We're on stomachs.
Yep.
Yeah. There's only two types of.
I was just looking at the wrong chart.
That's okay. Who else has a complex stomach?
Uh,
Think my favorite.
elk. Which is a
Marsupial.
ruminant. Oh, pseudoruminant.
Yeah.
Yeah.
Alpacas.
Is compartmentalized the same as complex?
Yeah, sorry. I should have wrote compartmentalized.
Marsupials.
Yes.
Why can't they use the words that I have in my head? Like,
Right.
it makes way more sense to be simple and complex
Right?
than compartment.
Compartmentalized. Simple compartmentalized, that doesn't make much sense. Okay. So, these are our types of stomach. And you probably already know this, but the proximal stomach does what? Or has what function? Storage or reservoir. And the distal stomach has what function?
Mucus.
What?
The mucus secreting.
Uh,
Are you talking about like the true stomach, or are you talking about like?
the true stomach.
Okay.
Break down.
It's
Yeah.
mechanical.
Yeah.
There are glands there. But it's,
I was thinking ruminants because they're my favorite.
that's okay.
Yeah.
That's okay. Is that okay? Okay. Alright, so the small intestines ends at what? What's the junction between the small intestine and the colon
Ileocecal.
called? Good. And there are 3 types.
Ileocecal, ileocoic, ileocecal-coic. This is only the first chapter. I should have had this week 1.
It's fine. Are you guys good with that? So who's ileocecal?
Horse.
Horse? Wait,
Ladder
no.
wolf.
Yeah, yeah. So horse and lagomorph
And
have
rodents.
ileocecal and rodents.
And rhinoceroses.
Rhinoceri.
Hey, I didn't either until last semester, like the day of the exam.
Didn't know what it was.
What a lagomorph was.
It's
Okay.
okay.
What has ileocolic?
Carnivores.
Carnivores. So dogs, cats, and ferrets. And who has ileocecal colic?
Pigs, ruminants, man.
Says us.
I'm glad you said that because I put my glasses on. I was like, that does not say man. But I can't tell what it does say.
Okay. Then we have 3 different types of colon. What are they?
Simple.
Okay.
Intermediate, complex.
Good. Who has a simple colon?
Carnivores and man.
Mhm. Who has an intermediate colon?
Pigs
Cows.
and ruminants.
And who has a complex colon?
Horses,
And
lagomorphs, and rodents.
yes,
But the horses is different than the others.
it is. Good job. You okay?
Yeah, I'm just old.
Okay. So a simple colon, what is the order of the colon? Real easy.
Descending, transverse, and descending.
Uh-huh. Does that make sense?
Yeah.
Mhm.
Okay. What about intermediate?
Descending spiral.
Uh-huh.
Descending spiral.
Uh-huh.
Good.
Transverse.
Good.
Distal.
Distal?
Yes.
Descending.
Descending.
Gosh, why cannot? Words are so
Close
hard.
enough for me.
Words are so hard for me. I'm sorry.
It's okay.
Yeah.
Alright, and then the complex stomach
Yeah.
for the horse. What does it start with?
Right ventral.
Yes.
Oh, you want the flexures too?
I want the flexures as well.
Okay, hang on. I gotta do this.
Okay.
Right ventral, and then make a little dash, and then
Uh-huh.
left ventral, make a little dash, left dorsal,
Uh-huh.
Mm-hmm.
then make a little dash, right dorsal.
Mm-hmm.
Okay, silly ponies defecated. So I have
Sorry.
the D, the defecated? Yeah. Okay, so we have the right ventral, then the I want to say splenic.
Sternal.
Sternal, okay. Then the left ventral, and then we have silly ponies. That is my pelvic.
Pelvic.
Then left distal,
Left dorsal.
dorsal. Then diaphragmatic
Uh-huh.
and right dorsal.
Yes. This is how I put it together to remember it, because I can't remember like right ventral, sternal, left ventral, pelvic, left dorsal. diagrammatic, because that's a lot. But if you kind of put it together like this, then you can write out however he wants it, and that makes it easier. Does that make sense, Kara? Okay.
I hate that I can't draw with my test.
We're supposed to get scratch paper.
It's supposed to.
I think it's done. Okay. So that's all you need to know about the gut anatomy. Do you want to talk about avian gut anatomy? Just real fast.
Do you want to do the breast? Oh, it's just transverse inducing.
Oh, yeah. Is that okay?
Yeah, that's fine.
Okay.
Dr. McCann doesn't like to talk to me.
Talk to you?
Yeah? No, I emailed him this morning about all the people freaking out because there's no notes on Moodle.
Why don't they just get them, like you can email that stuff?
Yeah,
He took down the
one for
chapters.
kids, chapters on Moodle.
Oh.
I wonder why he would do that.
What?
I have them all though.
I do too.
Yeah,
I don't
like-
understand. Like, what normal person didn't save
Doesn't
them?
have
Just
them.
email them now then across.
Yeah, just email them.
Instead of emailing
But he
him.
didn't.
I'm not doing it. Cause I draw over mine and stuff. Yeah. Yep.
People are gonna see what
Oh.
you're really doing in
Ex-
class.
exactly.
Just fine.
I can give you like,
If I had some blank ones, I would
I can
totally do
probably
it.
send you blank ones.
I mean, sure, if you want to do that, I'll send them to the class. I think that's a copyright infringement, but whatever. Mm-hmm.
I mean, he sent them to everybody, so I don't feel like it.
Yeah. Wow. We're paying to be here.
Yeah, I'm good with it.
Okay, so the bird gut. What does it start with that's different than others?
Crop.
So it has a crop. What is that?
Mm-hmm. Storage
Where is it at?
in the neck.
Mm. What organ is it in?
Esophagus.
It is in the esophagus.
That's in the neck.
That was not specific enough. So then where does it go?
We have a proventricular.
Proventriculus.
Which does what?
Mixes.
It's gland.
It is glandular first. Then where?
Ventriculus.
Which does what?
Mixes.
There you go. And it's
It's a
also,
gizzard.
what is it also known as?
Gizzard.
Mm-hmm. Yuck.
Love them.
My dad
Wow.
has them.
What is wrong with you?
Ugh.
Do you like them, Karen? Kiera? Wow. I
Whoa.
She's like, I'm not
totally.
gonna answer you.
Yeah,
until you
no
anyway.
joke, Kiera. That's so hard to say though.
Don't. It's hard. It's
Yeah.
hard to say. I'm sorry.
Yeah.
I'm sorry, my name is
Hard.
complicated.
Yeah, it is. There's like a whole extra syllable in there.
Kira?
Kira? See, that doesn't even sound right to me. It's like, you know, Simba and Kiera. No, I'm wrong.
Kiara.
Kiara. That's why I can't say it.
That's not my phone.
Okay, is everybody good with birds?
Yes. Yes.
Yeah.
Okay, you should probably also know that birds have
Really?
a double what?
Zika.
Good.
Cloaca,
And they have a cloaca.
which gives me eggs every day.
Yikes.
Fun
See?
fact, they are simple stomach animals.
Oh, did you find that out?
Yeah, he said that, and I was like, I kind of feel stupid now. They have nothing else that would tell me that it's not a simple stomach.
Yeah,
But they're also birds and they're weird and have extra things.
This is
so-
true.
Yeah.
Okay.
That's
Yeah.
fair.
So that is our review of chapter 1.
Woo!
Then we can
That
start
was
chapter
Quick
2.
review.
That's
Let's do
5
it.
minutes.
That was fast. How fast was it?
25.
Okay.
Do you wanna? Do you think we need to know about intramural glands of the gut?
We hit glands pretty hard in all the secretion ones, so I think if you know the difference between intramural and extramural, which is kind of self-explanatory,
Okay.
I don't think that will be too big of a deal. So what's an intramural gland?
Within the wall.
Mm-hmm. And then what's an extramural gland?
It's like on the wall.
It's outside and it has
Yeah.
pores that come in and drain into it. So what would be an extramural gland example?
Salivary.
Mm-hmm. What else?
Hepatic.
Mm-hmm. And
Pancreatic.
yes. And then intramural glands would be what?
Gastric.
Like gas. Yeah.
And
Goblet cells.
friends.
Goblet cells.
So, does anyone else like this is called the ampulla of Vader?
It's pretty cool.
What?
It's the
It has
ampulla of
pancreatic
Vader.
duct. It's also called the ampulla of Vader.
Ah.
Sounds crazy.
That was a scary sound. It's not now, I know what it is. It sounded like a dinosaur.
I mean, you could probably find a dinosaur egg out here.
Probably.
Honestly, yeah.
Dig into China.
Okay, so
Okay.
chapter 2 is about smooth muscle.
Yay.
There are two kind of classifications of smooth muscle. So it has to do with their innervation.
The multi-unit and
Mhm.
single unit.
So what is multi-unit and what is single unit?
Multi-unit is it like multiple cells functioning as one?
So it's actually kind of misleading with the name. Multi-unit means that there's
Oh.
every
Mm.
single cell is a unit. So they're each individually innervated,
I remember that now.
yeah, and they have no gap
Because
junctions.
single unit is like a single unit.
Yes, like every cell works as one.
Yeah.
So where would you find multi-unit?
Your eyeball.
So
Iris.
And then, so single unit has gap junctions and acts as a syncytium. Do you know what that means?
Still can't say that word.
Yeah, it's gonna be like as one.
Yeah, good. So there are two types of single unit smooth muscle. What are they? It has to do with how they contract.
Longitudinal and circular.
Not quite. That's what you guessed last time too.
Once it's wrong once.
Striated and smooth or something.
Nope, because we're just talking about smooth muscle.
Okay, what was the question?
There are two types of single unit smooth muscle and it has to do with how they contract.
Tonic and Phys.
Yay!
Oh my goodness,
Wow.
I did.
Nice.
I guessed that totally wrong last time.
You said it word for word of
I
what you
know.
guessed today.
Because I'm telling you, once it's wrong once, like I confuse myself. I'm like, which way is this actually supposed to be?
Write this down, Cody.
I did.
Write it down again.
Repetition is good.
Do you need a whiteboard?
I'm fine.
Are you sure? I have one and I have markers.
I'm good.
Okay.
On a test, I'll remember.
Okay.
Gail's like,
No, I did not. I did not. I
Mrs.
know.
Magic Mike over here.
I know you'll remember on a text.
Okay, so what is tonic smooth muscle?
It keeps the tone
Keeps the tone.
partially contracted.
Where do we find it?
That could be around blood vessels,
Mhm.
vasculature.
Where else? Maybe there's another place.
Your sphincters.
Uh-huh. And then one more. Think asthma.
Lungs.
Your airways.
Yeah. Airways.
Okay, and then phasic. What is that?
It contracts in a certain like phase,
Mhm.
like
So it has
peristalsis.
contract, relax cycles.
Yes.
So where would we find that?
Intestines.
Intestines. Where else?
Stomach.
Yeah. Where else?
Your swallow reflex.
It's a little specific. There's other places other than the GI tract.
Large intestine?
Other
It's a
than the
GI
GI
tract.
tract.
Oh.
Your heart.
No. Heart is not smooth muscle.
Oh, that's true.
Oh, never mind.
Cody doesn't have one.
Oh,
The
your
uterus.
uterus.
Okay.
I knew that.
And then one other place.
your bladder.
Your bladder.
Gosh.
Does this make sense? Is that clear? Okay. So now we get to talk about all our receptors.
Yay.
Our favorite thing, right?
Yeah.
What's the first word under phasic? I'm sorry.
Intestines. Do you need to scoot? Why don't you scoot up over here? Or right over there where you can see better.
I
Yeah.
might.
I
Can't
think
get
you
blind
should.
back.
Good thing bats are cute, Cody.
That's how
You're
rabies.
cute.
That's
What did you say?
That's rabies.
But I don't have rabies.
Are you sure about that?
Yeah.
Yeah. You're vaccinated.
I am. I am up to date on my
I am
vaccinations. I have a tag and everything. I don't.
Well,
People are like, why did you get a tag?
I was like, what?
What's wrong with you?
I didn't get a tag. Was I supposed to get one?
No, but they also gave me a seizure on my third.
Did they give you a city license?
No.
They give you a seizure on your third one?
Kira has.
I have a really bad
Problems.
vasovagal response to needles. I'm not afraid of them. I just pass out and have seizures.
Oh my god.
That's why I'm afraid of birth.
That's why I'm afraid
I
of
have good
fear.
reason. I'm like, I'm
Fair.
gonna die. I'm gonna die.
You're not gonna die.
Oh, I'm gonna die.
You'll probably be like one of those ladies that I want to be pregnant every 9 months.
Oh,
Once you're pregnant.
Ugh.
There are people like that.
I know.
Oh, pump the brakes.
Pump the
I want
brakes.
20 kids and I don't even want to have my wife pregnant every 9 months.
Yeah, I'm not a dog, thank you. Or a horse.
Okay, are we good here? Okay.
I don't want my own TV show. Everybody tells me that. I'm like, no.
I think you should have a TV show.
I don't want to get out of my life. That's what I should write a book and then turn it into a comedy show.
Oh,
Because
I'm writing a
everyone
book
would
about
be
vet
like,
school.
whoa.
I'm writing a book about vet school.
You're scared
Somebody
away.
That's how monopolies happen, Kale.
Right? Okay, so receptors that are on our smooth muscle cell. I'm gonna draw a smooth muscle cell over here. So, what is the one that is voltage dependent?
Fuck.
Okay, what does that stand for?
Voltage operated calcium channel.
Okay, I'm not gonna write all that out, but good job. So, what is it also known as? Think back to cardiac
Voltage
Phys.
L-type.
L-type
Oh,
calcium
that's
channel.
true, okay. We're going through our Rolodexes in our head and we're like,
And then he has one other name for it. It's called electromechanical coupled.
okay.
So, how does the L-type VOC calcium channel work?
It's voltage activated, so it
It is.
synapse.
It is.
Uh-huh.
So, do what?
What's it say, electrolyte?
Electromech for
Mechanical.
electromechanical.
Okay.
So
Someone explain it to me. So if it's voltage dependent, that means we have to have a depolarization. Are you good? Come over here.
I am.
Okay. Okay, so this is gonna be our little neuron, and it comes in and is going to cause a depolarization to occur. So, if our normal membrane is at like negative 80 millivolts, and we have depolarization happen, it's going to make our membrane go up to, say, negative 40 millivolts. And if that's the cutoff or the threshold potential, it's going to cause an action potential. Okay? So when we're at this negative 80, our L-type calcium channel is closed. But something happens during the depolarization that when it gets to negative 40, the channel's going to open up and calcium is going to be able to come in. Okay? Does that kind of make sense? Okay. Are you good with that? Okay. Right. What is our next kind of channel?
rock.
Yes.
Rock.
which stands for what?
Receptor operated.
Yep.
Calcium channel.
Yep. What is it also known as?
Pharmacomechanical.
So pharmacomechanical. And specifically, what type of receptor is it? Like we have ligand operated like channels, we have G protein, a couple of receptors. What is it?
G protein.
It is. Good job. Okay. Alright. So, can anybody explain to me how this one works?
Yeah, so there's a G protein underneath, a receptor comes and binds to, or a ligand binds to the receptor,
Mm-hmm.
and that induces a conformation change, allows things to happen, and then,
Perfect.
so this is the G,
Maybe.
go ahead.
I was gonna say, it ends up making PLC, right?
It does.
But this is a GQ?
Uh-huh. So the rock channels are specifically GQ because that's the only one that directly leads to calcium being released into the cytoplasm.
Because of the endoplastic, or the sarcoplastic,
Yes.
whatever.
Sarcoplasmic reticulum.
Yeah.
Perfect. So, how do we get from our ligand binding to calcium being released from the sarcoplasmic reticulum?
Okay, so you have the GQ
Mhm.
thing, so you got your GQ, which has the A thing,
The alpha subunit.
yeah,
What's the alpha? Where?
So this one would be like the alpha, and all it does is it is going to phosphorylate the beta and gamma subunits, and then they're going to make,
adenylyl cyclase.
no, that's GS.
Is it IP3 and tag?
Yes.
Ooh, that's
So
right.
we're going to make phospholipase C, which is an enzyme. It's going to take PIP2 and cause it to be cleaved into IP3 and DAG. What does IP3 stand for?
Oh.
I didn't-
Inositol triphosphate.
Yes.
And it's triphosphate.
What does DAG stand for? DAG is going to be diacyl glycerol. That's the only one he hasn't asked us yet, so it will probably be on the exam, because he's asked everything else, I think.
Diacylglycerol.
Yes. So what does IP3 do? From where?
The sarcoplasmic reticulum.
Yeah. And what does DAG do? Which one?
PKC.
Perfect. So this is your receptor operated calcium channel. Okay? Then there are two other types of channels that aren't quite as important, but they still play a role. Do you remember? It's SAC and SOC. Can you tell me what either of them mean?
Door operated calcium channel is SOC.
Mm-hmm. And what's SAC?
Stretch activated.
Good. So stretch activated calcium channel, and I'm going to talk about it first. So this is more like a conceptual thing for the stretch activated channel. So let's say you have all these little smooth muscle cells, and they're all here, because this is a little smooth muscle bundle. Okay? If this one has either a GPCR or like a rock that gets activated and it's starting to contract, it's going in because it's getting shorter. So that means it's going to be pulling on like this cell, and this cell. And then they in turn, there's going to be stress on these cells because this one's pulling on all of them. So what a stretch activated calcium channel is as the membrane is feeling that tension from the other cells stretching, there's going to be calcium channels that open in it, and then it contracts too. Does that make sense? There's not really a good way to draw that. It's just kind of, I don't know.
So you said the membrane contracts too? That's what else, like the muscle?
So
contract and then
the
that
membrane
causes-
is being stretched. The
Okay.
stretch opens up the calcium channel, causing that whole cell to contract.
Okay.
Because remember, in our smooth muscle cells, they've got these polar, are they called polar bodies? Not polar bodies.
Oh,
What's the word? Dense bodies. They're called dense bodies, and the dense bodies are stuck to the membrane, and then you have your actin and myosin in 3 dimensions that are attaching to each dense body. And so when it contracts, it's not contracting in 1 dimension like a skeletal muscle, it's contracting in all 3.
Okay, gotcha.
Does that make sense? Okay. And then what is the SOC? What's that stand for?
Storage operated.
So you probably don't have to know a whole lot about this one because we don't really understand it very well. But basically, once your sarcoplasmic reticulum is getting low on calcium, it sends some sort of message up to the membrane that says we need more calcium. So then the membrane will open up one of these other channels and let calcium in. We don't really know how it works, but it's so that the sarcoplasmic reticulum can restock.
Gotcha.
How's that? Okay. Now the hard part. How to contract smooth muscle. It's gonna be great. I'm gonna try and draw it better than I did last week.
It's not.
It's fine.
What's that sound? Do you hear this light buzzing?
I do.
Okay.
Oh, Dr. McKean? Turd.
Did he email you?
No. What'd you say?
An ash pool.
I'll edit that part out.
I forgot that was on the recording. What?
I think it's funny that he took it down. Like, why? What's the
Exactly,
point?
like-
Right. What does it matter?
Being sassy about something.
Okay. So in your smooth muscle cell, it's not like skeletal muscle. So in smooth muscle, you have an actin and a myosin, and they're already in like really close association. In skeletal muscle, there's troponin and tropomyosin that's wrapped around the actin, preventing myosin from binding it. In smooth muscle, they're already right here. The myosin has to be activated in order to be able to bind to actin. What is the region on the myosin that you have to activate, and what activates it?
The light chain.
20.
Yes. So what do we have to do to MLC 20?
We have to phosphorylate it.
Okay. How do we phosphorylate myosin light chain?
PKC.
What?
PKC.
Eventually. What is the enzyme that specifically phosphorylates?
Phosphatase?
Is it? What
No,
does
it's
the phosphatase
kinase.
do?
It's
It takes it off.
Mhm.
Phosphatase takes it off, kinase puts it on.
Yes. So myosin light chain kinase,
And we'll
because
see.
kinases phosphorylate.
And you have to have calcium calmodulin.
Yes. So you also have our little friend calmodulin. And it has to be activated by calcium to work. And what it does is when calcium is bound to it, it's gonna come over and help activate myosin light chain kinase. So just remind me, where is this calcium coming from?
The rock.
Mm-hmm. So here, and then you also have like a GQ, so that's our rock. So you'll end up having like IP3, bringing calcium from the sarcoplasmic reticulum, and then you have DAG, making PK what?
C.
C. Good job. Okay.
DAG. Diacyl.
Mhm.
Diacylglycerol.
Glycerol.
Glycerol.
There you go.
Diacylglycerol.
There you go.
Well, Alicia faced part of your problem. She could check there's 1, 2, 3 on Facebook.
Oh, that's nice.
Who did that?
Alicia. Johns.
That's her job.
Well, yeah.
Okay, so myosin light chain kinase. Is it active when it's phosphorylated or dephosphorylated?
Dephosphorylated.
Okay, good. So that means it's kind of going back and forth with its inactive phosphorylated self. So what converts it from the inactive phosphorylated to the active dephosphorylated state?
Myosin light chain kinase phosphatase? Or is that exon?
No, that's right. So this is kind of what we didn't talk about last week. So PKC indirectly makes myosin light chain kinase inactive to the active form. So how it does this, get this, it activates an enzyme called myosin light chain kinase phosphatase, which, what's a phosphatase do?
Dephosphorylate something.
Yes, so it is going to dephosphorylate myosin light chain kinase, making myosin light chain kinase active. Okay? So that's kind of a little bit of an extra step. Okay? So, what activates myosin light chain kinase? What kind of comodulin?
Calcium comodulin.
There you go. And then what turns myosin light chain kinase from the inactive form to the active form?
MLCK phosphatase.
Which comes from where?
DAG.
Which comes from what?
C.
GQ.
PKC.
Yeah,
Oh.
you're both right.
Oh.
So, like DAG makes PKC, which then makes myosin light chain kinase phosphatase, which comes from what kind of receptor?
GQ.
GQ.
Perfect. Which is also known as,
Rock.
which stands for
Receptor operated calcium channel.
yes. Are you good with this? Okay. Alright. So now we're going to kind of branch into how relaxation works. So do you understand how this gets us to contraction? Okay. So now we have a GS. Somebody remind me what GS does.
Stimulates adenyl cyclase. Adenyl cyclase.
Adenylyl cyclase.
Adenyl
And then it makes CAMP.
Yes, which makes what?
PKA.
Perfect. Okay, so we're gonna leave that there for a minute. If we have active phosphorylated myosin, how do we deactivate myosin?
Myosin light chain phosphatase.
Perfect, because we are removing the phosphate group from the MLC20 region of myosin. Is that clear? Okay, is myosin light chain phosphatase active when it is phosphorylated or dephosphorylated?
Phosphorylated.
Okay, so we're gonna put a active phosphostate. And then that stands to reason that sometimes it's going to be in its inactive state, which would be phosphorylated or dephosphorylated.
Dephosphorylated.
going to come over here and remove that phosphate group. So, what enzyme is going to take it from its inactive dephosphorylated state to its active phosphorylated state? I asked that badly. Hang on a second. Okay, so we have pKa. What other protein kinase have we not talked about yet?
Hmm.
We've talked about C, we've talked about A.
Mm-hmm.
Who else do we have? Okay, where does protein kinase G come from?
G, A, and P.
It's gonna be CGMP.
C, G, and P.
Uh-huh.
Okay.
And then where does that come from?
Guanylyl cyclase.
Mm-hmm. Which is from a GPCR, and what is the ligand that would bind to it?
GI.
Think of a gas.
Nitric oxide.
Yeah. So nitric oxide relaxes smooth muscle, so it's going to bind here. It's a GS as well, but instead of adenylyl cyclase, it's going to make guanylyl cyclase, CGMP, and PKG. So we have that. So PKG is actually going to be what activates myosin light chain phosphatase. Okay? Okay. Sorry I asked that, kind of weird. And then if we are turning relaxation on, we have to turn contraction off because it doesn't make sense for them to both be going at the same time. So the easiest way to do that is to convert myosin light chain kinase back to its inactive form. So if we're kinasing something, that means we're adding a phosphate. So who do we have left that's not already phosphorylating something?
PKA.
Yes. So pKa is going to come and turn myosin light chain kinase into its active phosphorylated form. Do you have questions?
Yeah. Can you repeat that last part?
Okay. So pKa
Uh-huh.
is going to phosphorylate myosin light chain kinase and deactivate it so that we can stimulate contraction.
Relaxation.
Relaxation. So, sorry. Words.
So it's phosphorylating myosin light chain kinase.
Yes.
PKC also dephosphorylates MLCP, right?
Mhm.
Okay.
So think of MLCP, I think it's either a tetramer or in 3 parts. So here is like our little active phosphorylated area and it's working. So it's causing relaxation. So what PKC is gonna do, because PKC is a kinase, so it's adding a phosphate group, it comes and it's gonna phosphorylate one of these other subunits, which in turn keeps this subunit from working.
Okay.
So that's why, like, myosin light chain phosphatase is inactive when it's dephosphorylated, because it's this phosphorylation that we care about.
Gotcha.
So even though PKC is a kinase, it is still kinasing, but it ends in a dephosphorylation.
Okay.
Okay? So that's where some naming confusion comes about. And then I think there's one more we're missing. No, yeah. Okay. So what are 2 things you need to do to cause contraction? 3 things.
Inhibition, and you need to stimulate contraction. So
little
inhibit
broad.
relaxation.
Yes.
That's what I meant by that.
Okay.
And then stimulate contraction.
Okay. What's the first step to stimulating contraction?
It would be the VOC, and then that
Mhm.
whole pathway that phosphorylates the myosin.
Okay. So to contract, you
You have to get calcium.
Get
come
calcium.
on.
Okay.
get calcium. So we're going to do that with box and rocks for the most part. Then you need to activate myosin light chain kinase. So what are the two things we use to activate myosin light chain kinase?
Calcium carboxylate
Mm-hmm.
and MLCK phosphatase.
Mm-hmm. And where's MLCK phosphatase coming from?
PKC.
Okay, and then the third thing we need to do to contract is we need to inhibit relaxation. How is that done? So do you want myosin light chain phosphatase to be
PKC.
active or inactive?
Inactive.
Okay.
So it's a PKC.
Inactive MLCP via PKC. Okay? So these are your 3 steps to get contraction. Does that make sense?
Mm-hmm.
What questions do you have?
So,
I can see you questioning.
I know. Calcium, how much do I get?
Mm-hmm.
The MLCK phosphatase, and the PKC.
So PKC is making MLCK phosphatase.
Mhm.
So phosphatase dephosphorylates. So it's going to dephosphorylate this inactive myosin light chain kinase, and that is going to turn it on, because myosin light chain kinase is
It's
only
and
active
it's
in the
dephosphorylated.
dephospho state.
So you just need myosin light chain kinase.
Yes.
You need to make sure it's in your active form.
Yes.
I think that threw me off as well.
Sorry.
No, it's fine, because he just puts myosin light chain kinase is, but it
Yeah.
needs to be in its active form.
Yeah.
Active.
And then, okay. So then if you understand how to contract something, relaxation is going to be the opposite. So how do you cause the smooth muscle cell to relax? Give it
Take
a
away
mirror.
calcium.
Yeah, perfect. How do we do that? We haven't talked about that yet. Just so we
Take
stretch
away
your
calcium?
minds. Yes.
You close the channels?
Yeah.
Mm-hmm. So we're gonna change our voltage, maybe.
Mm-hmm. And then you're gonna store it again.
Yes. And then what if we pump it out of the cell? Does that sound like a good idea?
Mm-hmm.
I think that's a good idea. So that is going to be called an NCX channel. And what it does is it pumps calcium out. And who comes in, do you think? Sodium, because sodium always wants to be inside the cell and we never let it. So we only let sodium come in if we can send calcium out. So this is getting calcium out of our cell, so who do you think is going to be stimulating this little pump to be working? It's gonna be 2.
I get that.
Okay, so PKA from our GS
Okay.
is going to be stimulating that because it's already over here turning off contraction. And then PKG is also gonna be stimulating our NCX channels,
That makes sense.
because it's already over here activating myosin light chain phosphatase,
Okay.
okay? So we're gonna remove calcium by changing our voltage, by opening an NCX channel, and getting calcium back into the sarcoplasmic reticulum. And it's actually gonna be PKG that does that part too. But I don't really have anywhere to draw that, so I'll just have to take my word for it. So if our first step to relaxing is removing calcium, what's our second step? If it's
Inactivating MLCK.
perfect. How do you do that?
PKA.
Yes. Which is just going to put a phosphate group onto MLCK, make it deactive. Is that cool?
Cool.
Okay. What's the third way to cause relaxation? Or the third thing you have to do?
Inhibit contraction.
Inhibit, stimulate relaxation. Because inactivating MLCK is already inhibiting contraction. We just switched the wrong thing here. So it's gonna be stimulating relaxation.
Okay.
Because when you inhibit an inhibitor, you stimulate.
So how do we stimulate relaxation?
With MLCP.
And how do you turn MLCP on?
PKG.
PKG.
Yes.
Oh. Okay, can you explain to me the green, I think green, the green arrow that's coming all the way across to the MLCP again?
Yes. So is MLCP active when it's phosphorylated or dephosphorylated?
Dephosphorylated.
Okay.
What?
No, that's
Yeah, I
wrong.
was gonna say.
Dang it!
MLCP.
MLCP, phosphorylated.
So it's active when it's phosphorylated. So we're just gonna pretend that this is our little phosphorylated active MLCP. So PKC is over here, and it's your great stimulator of contraction. So
Okay.
part of stimulating contraction is inhibiting relaxation. That goes hand in hand. So while it's
Agreed.
doing all this stuff, it's also got its little nose over here in myosin light chain phosphatase saying you can't do what you want to do. So the active form of MLCP is dephosphorylated. If I tell you that protein kinase C is dephosphorylating myosin light chain phosphatase, how is that possible? Because kinases don't dephosphorylate. So what it's actually doing is your protein kinase C is going to add a second phosphate group onto a different subunit of myosin light chain phosphatase, and then that subunit causes a conformational change of the whole thing that ends up kicking off this phosphate group, making it the dephosphorylated inactive form of myosin light chain phosphatase.
Okay.
Would you coming and drawing it out help? Would you saying it back to me help?
So I'm not wrapping my brain around how when you kick that phosphate out, how come that doesn't keep it in the active form? Because there's still a phosphate there.
Okay, so
This would help me maybe.
Okay.
Is there like a specific place that it has to be? Okay,
Yes.
that makes everything better.
It has like three.
so
It has to be on a certain one for it to
Okay.
work.
Okay,
so there's
I'm good
3 units, and this is the only unit that can dephosphorylate myosin.
then. Okay,
And if that part of the unit isn't phosphorylated, the whole thing doesn't work.
okay, got it.
Is that better?
Yeah,
Okay,
I have a pea brain, so.
you're good. You're doing good. All right, so somebody want to walk through this?
I'm gonna have a question, just let me catch you
Okay,
real
you're
quick.
good.
It's chapter 2, right?
Mm.
Yeah.
Okay.
So PKG stimulates relaxation.
Yes, because it comes from where?
Nitric oxide.
And what is nitric oxide's great thing that it does?
Inhibition. Stopping bad stuff. So nope, that's all
Okay,
I think.
you guys good on this? Any other questions? Speak now, forever hold your peace.
Diacylglycerol.
Uh-huh. What does AC stand for?
Adenylyl cyclase.
Mm-hmm. What does IP3 stand for?
Ooh,
Tall track
yeah,
phosphate.
that's one I can't even remember.
What does GC stand for?
Oh, guanylyl cyclase.
Yes.
Oh.
What
I
is
thought you were talking about like GQ, GS. I
Oh.
was like GC.
I know, that's where my mind went too, I was like,
You're like, I know we haven't talked about that. What does NCX stand for?
Sodium calcium channel.
Exchanger.
Yeah,
Okay. Alright, I think this is good. We're gonna have to keep rolling.
yeah.
How long do we have the room for?
12:25.
What time is it?
It's 11:45.
Oh, okay. I don't think we're doing bad.
We could probably go recheck it out if we need to. I don't think there's anything here.
Yeah,
Okay, is that the end of chapter 2? Just a question.
I believe so.
Okay. I'm just
I think I bumped it a couple times, I'm sorry.
No.
Put my feet on the bottom of the chair.
Dear, okay.